Thanks, Beverly. Thank you, everybody, and thanks for the opportunity to speak to you today. I come from the Tech Transfer Office at Stellenbosch University. We often look at the research pipeline of the university and look at how those innovations and technologies can be taken out to society through commercialization. Okay, so today I want to talk to you about publishing discovery science and about the case of CRISPR Cas9. Okay, let's see. There we go. So I'm going to talk a little bit conceptually about public science versus private science, and then key events in the development of CRISPR Cas9 and all the way to the gene editing tools. And then I want to talk a little bit about this case and its implications for research institutions. I'm not going to give you the biotech lecture. I don't understand it very well. But I am going to try to look at how this case affects a lot of our decisions in society around technology transfer, and also how some aspects of how science communication possibly needs to intervene to help society understand and help institutions understand the value of the technology and how to ensure that benefits society. Okay, so long ago, when everything fitted neatly into boxes, there was public science and there was private science. Public science was very much as a public good, like schools or roads or public health care. It had very much characteristics that it was done in public research institutions, mostly with state funds or with philanthropic funds, published and shared very, very freely, and the purpose was for public good. It was to push the boundaries of knowledge. Public goods generally have the characteristics that they're not depleted when shared. Once made available to the public, nobody can be easily excluded from it. And then the incremental cost of an additional user is almost zero. Very differently, private science was done in private firms, in businesses, and it was done to give that business a competitive advantage. If it was done in a public institution, it was often paid for by the private company, and what was very important is that we needed a mechanism to protect that private science so that it could give that investor some kind of competitive advantage in their business. And that is how intellectual property rights have been derived, and patents, copyrights, and designs have been derived around that, so that we actually protect that intellectual property so that whoever invested in developing that intellectual property can gain some kind of monopoly advantage in the business environment, okay? So it allows someone to earn recognition or financial benefit from what was created. Now, by striking the right balance between the interests of innovators and the wider public interest, the intellectual property system aims to foster an environment in which creativity and innovation can flourish, okay? But what happened in the 1980s is that the public science and private science collided. In America, they published the Bayh-Dole Act. With that, public universities could suddenly protect their intellectual property, file patents, and then license out those patents. In South Africa, we had our Intellectual Property from Publicly Financed Research and Development Act, which was put into place around 2008. Okay? So also we protect intellectual property from our universities, from our public research institutions. We look at ways to protect them and then commercialize them. Okay? So now let's consider what's been happening with the CRISPR case. Okay? With CRISPR, the case gives us a clear example of how we've moved from discovery science into the application science arena, and how patents have been used to protect that commercial opportunity. The priorities of researchers their research institutions, businesses, and commercial entities that are involved change along the way. How does this affect how it's communicated, and how does it affect the trust of public within that movement? The benefits to society and how it plays out is dependent on how this process is managed. Now, one of the researchers who was involved from the very beginning has been George Church, and he says it's good enough, but it's not all for the good. So it's important to look at who's going to be benefiting down this process. So what is CRISPR? And in the discovery science, what it was, it's clustered, regularly interspaced, short palindromic repeats, okay? Basically, CRISPR was a mechanism that was discovered in prokaryotes, and it was helping the prokaryotes to protect from virus invasion. So it was almost an immune system for prokaryotes, which is a group of bacteria and archaea. Okay, so they're not eukaryotes, so they're prokaryotes. Sorry, the biology lesson. Okay, so basically it has a mechanism where it identifies viral invaders and then destroys those viral invaders by interfering with their RNA or DNA. Okay, so the discovery science started all the way back in 1987 with a paper from a Japanese university. It wasn't even called CRISPR at that time, but they started noticing regular repeated sequences. And nobody knew what it was. It was something that was discovered, worth publishing. Carries on into the 90s and into the 2000s. There was a team in Portugal that also discovered this, and they helped coin the term CRISPR later on. Then you get more and more publishing around this, but you can see from about 2013, you only had about 223 science articles published about it. Moving to 2016, when you're starting to see 2,225 articles published in it. And then moving to 2023, 8,652 articles. In between that timeline, some key events happen. For instance, in 2008, there was an attempt to start protecting this intellectual property, and there was a patent filed by Sontenheimer and Ravini. And they made a discovery and tried to patent it. But when it was filed in the United States Patent Office, that patent was never granted because there was indication that it was not novel. There was some science, there was some indications that CRISPR was happening, but there was no application. And critically, for a patent, it needs to be novel, it needs to be non-obvious, and it must be industrially applicable. This patent didn't have an application because nobody understood it by that point. Okay, discovery science, very much in that area. However, moving out of 2008, moving towards 2012, 2012 is when the first real patents showing how the discovery could be manipulated into an industrial application. So that's starting 2012 when the first really important patents get filed on CRISPR. And in 2020, we get the Nobel Science Prize for Doudna and Charpentier, and it's the first time that the Nobel Prize is awarded to two women. Okay, so, and then going beyond that, and in the media these days, it's the big issue is who owns the patents, what are the patent offices saying, what's the big fight about it, and all the public relations spin around who owns what patent and who developed what. And it's starting to get really ugly. Even some of the scientists, even George Church, who was one of the original scientists, has noted that this is really getting very, very ugly. Okay, so So from that discovery science era, we start moving into the gene editing era, right? So we understood now what the system does. Now the scientists have developed what can be manipulated in the system and how it can be applied to eukaryotes to do very specific, very precise gene editing to help prevent certain diseases or to improve certain crops and certain yields, and it has been manipulated now to actually do very, very specific gene editing. I'm not going to go into all of that, but these are some of the key events in the published science about what happened. This is a description of how that CRISPR Cas system works in terms of you first manipulate the Cas to actually start picking up the DNA or RNA that you want to change, and then you put it into the system, and ultimately you're going to use the cell's own repair mechanisms to actually change the genes to what you want it to do. So that is now what has been patented, and that is what's been used to actually create those three companies. Those three companies hold the patents and license them out in order for them to be used in various applications, from industrial biotechnology to human health to environmental applications. So the scientists on that side are participant in some of these companies, and those companies then license out for various applications to be used in industry. That's a really nice interactive diagram if you want to go to, and it's from Jay Cohen's article. Okay, so what this has shown us is a very clear distinction in this movement of discovery science to application science, and then the protection using patents of that application science. But all the public is really getting involved in is hearing about the disputes about the patents. So we need to start thinking about how and when the discovery science communication starts taking place and how and when it is managed. From a research institution perspective, it's also very important for us to start thinking about what to publish and what to protect. But what's getting more scary, it's about when to publish and when to protect. If we think about all the way in 1987, the first discovery science was done. And huge strides were taken in that time frame between 1987 to 2012, when the first patents are published. And all the research institutions that were involved, various countries, various researchers, would they be getting benefits back from all that initial discovery science? What is this going to do about researcher behavior? Are researchers going to be rethinking about when to publish? Are researchers going to be holding on to their research, hoping to patent first before they publish? Sometimes you don't get grants which will fund all the way from discovery science to application science. So what's going to happen and who's going to have access to that information? So just to round up, accessing funding and who is able to access funding, developing countries, where do they fit in? Knowledge created in discovery science, how is that managed within developing countries and developed country partnerships? Who owns background IP? How are we going to manage benefit sharing across those different research consortia? So those are the questions to think about. There's some references, and that's it for me. Thank you.